In Module 1, we studied the origins of life and how different plants and animals evolved on Earth, therefore drawing connections between shared derived characteristics of various organisms through the interpretation of phylogenetic trees. Fossils allow us to study these derived characteristics, and by looking at fossils from various time periods, we can build evolutionary relationships. The evolutionary relationship we want to analyze in this video is one of a nerve cord present in an ancient panarthropod. Recently, fossils were found of the species Changjiangocaris kunmingjensis, a shrimp-like organism that resided in South China about 520 million years ago. The distinctive vertebrate structure consists of condensed ganglia along a ventral nerve cord that extends throughout its entire body. With the discovery of its segmental and intersegmental nerve roots, scientists found that this structure is common in some of its relatives today, but absent in others. This raises questions about how the ventral nervous system evolved in Panarthropoda and where it changed, with the goal of allowing fossils to help understand the evolution of arthropods. Before we begin to look at the data of the experiment, there are some terms you need to know. The ventral nerve cord is a bundle of nerve fibers which is essentially an invertebrate's equivalent of the central nervous system. The ventral nerve cord is similar to the spinal cord of mammals. In most cases, this nerve cord runs ventrally below the gut and connects to the cerebral ganglia. Ganglia are large numbers of neurons aggregated into clusters. Lastly, a phylogenetic tree is a branching diagram that depicts the evolutionary relationships among species or other taxa. To analyze the data, various powerful cameras were used to collect pictures of the fossils. Then, using a microscope, scientists analyzed the chemical compounds of each fossil and performed a phylogenetic analysis. Essentially, they attempted to infer the evolutionary history of the creatures in question to see where exactly they fit on an evolutionary tree by examining their anatomy and comparing them to modern organisms. In this way, they would be better able to understand the way in which the central nervous system evolved. This species shares a common ancestor with arthropods and velvetworms as shown in the phylogenetic tree. The large number of fine nerves found in C. kunmingjensis makes the species similar to velvetworms, which also have a large number of fine nerves. However, this number is significantly larger than the number of fine nerves found in arthropods. The nerves were found to extend from the nerve cord in regular intervals towards the underside of the body, which is also more similar to the structure found in modern worms than in modern arthropods. As shown in this phylogenetic tree, the phylum Onychophora, velvetworms, retains these characteristics while the phylum Euarthropoda, modern arthropods, lost these characteristics. A conclusion reached by the study was that simification was an important part of the evolution of the nervous system. These findings are important in that they give insight into the evolution of the central nervous system. This study exemplified the use of phylogenetic trees as well as demonstrated concepts of early development of the central nervous system in animals as we have learned in Biology 1520.